Hi, this video introduces you to Working Group 5 of QUAREP-LiMi. We are tasked with identifying the simplest, most robust method for tracking the resolution of a light microscope system. We are estimating resolution by looking at the resolved size of a diffraction-limited spot, known as a point spread function, or PSF. This can be monitored on the same microscope over time, helping the user identify any potential problems. This does not mean a minimum resolving distance of two objects, but it is still a reliable and consistent measurement of the resolving power of a microscope. Since we decided to use such a measure to follow resolution, in line with the published confocal ISO, we have tested different samples to identify something that would allow us to record the data we need, and we're working to identify the metrics that will allow us to best characterize the data. We deliberately aimed to do this for point scanning and spinning disk confocals initially, so as to identify the crucial parameters required for robust QC. We focused our attention on, first, sample preparation, second, image acquisition, and third, image analysis. All of this work culminated in the first version of a protocol's I/O manuscript, which gives a method for creating the bead slide, imaging it, and the metrics to capture from the analysis. We tested some fixed and printed substrates as well as beads, and settled on 175 nanometer green beads based upon brightness, stability, cost, and accuracy. The protocol gives detailed instructions for preparing a robust sample with these. We optimize the imaging parameters and next give the details for this in order to capture the data in enough depth to provide the metrics we required. This involved investigating signal to background, voxel size, as well as other parameters strongly affecting the image, such as lens cleanliness, sample entropy, and others. Finally, we investigated the output from various software analysis packages and described in the protocol which metrics should be observed over time. Our current work is focused on 4 activities. First, writing a peer-reviewed manuscript based upon this work. Second, looking in more detail at the impact of signal-to-noise ratio on the resolution estimates. Third, investigating in more depth the best fitting method for determining the image bead size. And fourth, exploring ways of easily identifying potential aberrations from these fits. In future, our immediate aims are to expand this work into wide-field microscopy, and then building this work into a new version of the published protocol. Later, we intend to establish methods suitable for other immersion media. Then, we would also like to look at the best method of monitoring system resolution on other microscope types, such as single molecule imaging, SIM and STED, as well as light sheet systems, which may also bring in the possibility of testing and monitoring the differences of point spread functions with imaging depth in this microscope system. If you're interested in telling us your preferences for our work focuses, please complete the survey we've released in line with this meeting. Thanks for listening, and we'd welcome you if you wish to join us in Working Group 5.